My definition of innovation is when you got something that nobody wants. That's how you know you got something. Anybody in science actually knows that innovation implies newness, but there's nothing really that's new anymore. Taking many different disparate things and putting them together in a way that they're used for something that you didn't think of before. So they may be things that are quite mature, but put together in a new way, they're greater than the sum of their parts. Think back to the 1960s. The culture at that time was very pro-science. There was high regard for people going into the sciences and engineering. So a lot of us were just following the cultural zeitgeist.
Back in the old days at SCIEX, it's a completely different organization. SCIEX was a very small company where it was really, really driven by the scientists.
These were people of extremely highest caliber. Bill Davidson, Bruce Thompson, and another fellow, Don Douglas. That culture of basically scientists running the company was strong. Well, this might come as a surprise, but it was more about enabling everybody to do everything they could to violate every rule of work-life balance. The divorce rate was pretty high in the R&D group at those times, but frozen TV dinners were free to get you through the night and weekends. But this is exactly the kind of thing that groups of young scientists thrived on.
SCIEX at that time was known for its mobile detection system, so they would go to disaster sites.
We developed the first single quadrupole dedicated atmospheric pressure ionization mass spectrometer, designed it to be mounted in mobile laboratories, but what immediately emerged was an understanding that the complexity of polluted air was far greater than we'd ever imagined. That led to the development of the first commercial triple quadrupole mass spectrometer, and this iconic instrument was called the TAGA. It was bomb-proof. And
there was a big train crash, and most of the town or city called Mississauga was evacuated because of the contents of the rail cars. And so SCIEX sent in a mobile monitoring system through the Ministry of Environment in Ontario, and they monitored where the plume went. So they would drive around and they would look for the traces of the noxious materials, and they mapped out which parts of the city had to be evacuated.
Things like going after train wrecks with hydrocyanic acid spills, and one of the big ones, the Love Canal disaster in Buffalo, New York. It was a situation of chemical waste being buried, neighborhoods being built on top of them, and carcinogens leaking into the basements of these homes, and cancer rates skyrocketed. And this TAGA instrument was one of those on-site identifying and quantitating what these materials were. And a lot of them were some of the worst actors in existence, the dioxins.
Back in those days, if you were nearby, you wanted to know that it was either safe to stay or not safe to stay. And in most cases, it was not safe to stay.
We as a population, as humans, have become conscious of the world around us in terms of environmental pollution, for example, food safety, water quality. The science has been developed in parallel with us understanding how technology can be used to benefit us from as a population. The way we communicate, that's got faster. The world's got faster. The computer systems have got quicker. What's it going to look like in the future is the other question.
It turned out that developing all the technology around successful atmospheric pressure ionization was very prescient and would pay enormous dividends in the future. If there was any mistake we made, it was working on an advanced technology for all the wrong reasons. We were focusing on it primarily for, like I mentioned, pollution control, but its true value emerged 10 years later when we realized that it could be used very powerfully as for a method of liquid sample introduction into the mass spectrum instead of gases. This opened up the biomedical field.
It's always good to identify things, but one always needs to know how much there is, because it's always easy to detect something. But what does it mean at the end of the day? We always need to turn it back into a quantitative answer. Without quantitation, it's hard to actually say to people if it's important what you've detected or not.
There was an enormous, around the world, research activity in trying to find a way to couple two fundamentally incompatible techniques. That being mass spectrometry and liquid chromatography. We called it the liquid phase, gas phase dilemma. Cracking this problem, everybody knew was the holy grail. We thought we had something, but you're never really quite sure. We, as scientists, had to go to our joint venture partners, and the response was, this was on the lunatic fringe.
The API-3 was essentially a cobbled-together instrument based on our earlier triple-quad technologies of the TAGA 6000, where it now had an electrospray ion source, as we called it, ion spray. It was remarkable in that I think it was the first electrospray LCM-SMS system on the market. There were a lot of naysayers in the company who didn't think it would sell very well.
And what we did was went directly to people in our customer base, if you will, but in all honesty, what we were showing them looked like something that came directly out of the age of stone knives and bearskins. To all their credit, they saw through the superficial aspect. They supported us to a level of purchase orders. That put pressure on us. I mean, it got so crazy. I looked at a couple guys and I said, you're good at carpentry, I'm good at carpentry. We started to build this machine out of wood.
People loved the instruments. They were incredibly sensitive. They solved problems that had existed for decades, and that was how to measure analytes in an LC flow reproducibly and with high sensitivities and signal-to-noise ratios that just were not possible before.
One of the challenges associated with the API-3 is even though it was a very sensitive system, every 60 hours we had to recycle it and repump it down 24 hours. So you would actually have a long wasted time when you were doing the API-3 analysis.
So we had to come with a way around it. That led to a discovery of an entirely new physical principle. It was the principle of collisional focusing. And this allowed us to pump the gas away and focus beams through narrow slits and differentially pump so that we could use more conventional turbomolecular mechanical pumps instead of cryogenic pumps. To this day, 99 if not all, atmospheric mass spectrometers on the planet today utilize that principle. There was an enormous explosion in the entire field of mass spectrometry to the point where it essentially converted almost overnight from a vacuum-based ionization technique to atmospheric. The driving force then became sensitivity improvements. And this, of course, was coming from the people using the equipment.
It was changing. You were used to your HPLCs running all night. Suddenly you had this new piece of software and hardware. People had to understand what the data was showing, but it was the speed of that. And that's why they adopted it. So instead of getting a result from one sample in 18 hours, you were getting it in minutes. When you look in the bigger picture of that, it means you were getting through all the studies faster, people were getting the results quicker, so they were making faster decisions, and it was helping them move the whole industry on.
SCIEX always seemed to have that little bit of extra edge, and they would always come out with, we've got a new instrument, it's 10 times more sensitive. And you sit there and go, how the hell are they doing it? So you try and compete against that, and you always found that they're just ahead of the curve from everybody else.
So we knew for sure that the days of single analyte was kind of out of the equation now. People wanted to do more and more analyte quantitation simultaneously, and that's what led to not only the development of the Turbo V with respect to ionization, but a lot of improvement in the ion optics.
The Turbo V was one of the first, if not the first, ion source where background noise was reduced because of the design of the ion source. I think our competitors reacted to us more than we reacted to them because we were the leaders at the time and still are.
Trying to drive to lower and lower limits of quantitation. It was a frantic research effort on everybody's part in the industry, putting things like bigger vacuum pumps. That was a model we called the API 3000. Quickly following on the tail of that was trying to figure out how to get more, to create more ions inside the ion source at atmosphere. That led to the API 4000.
By the time they came out, I would say the SCIEX was really entrenched in the industry, and it was the gold standard.
That was an enormous jump in sensitivity at conventional flows, almost 2 orders of magnitude in many cases.
The increased sensitivity is really what excites me about it. It's the ability to see ultra trace levels of particular molecules and ions that you didn't know were there.
It became clear to us that the end was in sight. And if you look at the graph of sensitivity improvements over time with each successive model, you see it reaches an asymptote. It's still going up, but it's going up slower. It's not over yet. There's still more to be gained. But that clued us in to the fact that we had to look elsewhere.
In the early 2000s, other companies were starting to catch up with us in terms of sensitivity. The gold standard had kind of moved away from the API 3 to the API 300, the 3000, the 4000. The 4000 was great, but the ability for us to offer a model of every one of the triple quadrupoles with Q-trap functionality gave us a huge, important differentiating feature that our competitors did not have.
SCIEX has traditionally been the leader in triple quad technologies. We're the first ones to bring the LCMS to the mass market. So the challenge is continuing to innovate, making sure that we have the next best thing that will enable our customers to make the discoveries that they need to do in order to progress their research or help make people's lives better. So we're always looking to find the next greatest thing for our customers to make them successful in their jobs.
Every biological system is incredibly complex, more complex than we can even imagine. The business of taking a large cohort of patient samples and getting relative quantification has always been extremely difficult. I think a game-changing technology like SWOTH MS had to come along.
So SWOTH acquisition came about from understanding that customers wanted to quantify many more molecules than realistically they could build mass spec methods for. The studies that biologists want to do are not just 3, 4 samples, 5 samples, they're getting to the hundreds and thousands of samples. And from there we had to come up with strategies on how to process that data fast enough and enable the end user to take those numbers and turn them into something that they really understood.
The Stoller Biomarker Discovery Center was developed in order to find biomarkers of, say, risk of having a certain cancer. We need to have signifiers, biomarkers of response to therapy or diagnosis.
SWOTH is our preferred approach for many reasons. It has high reproducibility, high sensitivity, very high dynamic range, and the most important thing, we can get very efficient and good proteomic signature maps in almost a couple of hours.
I think the speed and sensitivity of the instruments has increased a great deal. This generates a lot of work in terms of informatic analysis, but it also generates a lot of opportunities as well.
For PhD students and also for research technicians that worked at the Stoller Center, I can see a great future in proteomics.
We've had quite a lot of different diseases. We've had Alzheimer's, we've made some good progress with that. We've had ovarian cancer. I've worked with some heart disease. And mainly we're looking for a variety of different biomarkers, but they can tell us a lot of different things. So really that's the main bent of the study, is to build up these almost like fingerprints for each person and for each disease, so that we can predict how people are going to respond to illnesses, what diseases they're going to get, that kind of thing. That's the main focus of the research.
You could have a healthy diet for all your life and you're still prone to cancer. That's still misunderstood. It's not well known. The technology we can develop helps to understand at the submolecular level what is actually going on.
There are more drugs coming out and faster. The drugs are better. And ultimately, if we have that scenario, people are going to get the drugs and we're going to save lives.
So the technique SWOF allows quantitation of metabolites, proteins, lipids. It allows us to understand what actually is a snapshot of a body. And if you put that together in a longitudinal time, you can understand how the body changes. If we can detect cancers early, then in reality, you won't lose your parents early. You won't lose your children. And we might be able to actually make it a better place because of that. Quantitation is the future. It's not static understanding what's in a sample anymore. It's understanding that quantitative changes are really what makes biology.
It is one of the most important things in the world to know how much of a variety of substances are around. Just it affects our entire lives.
In my mind, Generation Quant means that generations of scientists have taken the technology and moved their science forward. Mass spectrometry is a technology that's so unfamiliar to the general public, but it touches them in so many different ways. Over the generations, people have driven that technology forward to deliver where we are today and where we're going to go in the future.
Mass spectrometry is always coupled to an LC device in 99% of the applications out there. And that's where there's going to be probably a huge opportunity for us as a company to potentially take advantage of the added sensitivity that we've gained over the years and remove the LC. Not only would we gain significantly in terms of analysis time and throughput, but it would also greatly simplify and broaden up the number of applications or our user base. We definitely benefit from that.
What we're doing today, if we looked at it 20 years ago, it wouldn't be possible because the mass spectrometers weren't sensitive enough. It addresses all of the bottlenecks in quantitative analysis, not just one. Part of the reason for this is because sample preparation is eliminated and much of the variability in measurements comes just from that. We think that this technology, it's certainly going to change things quite a bit.
It's incredibly fast, the data is incredibly reproducible, and it gives very, very fast quantitation on a huge number of analytes. And it's adaptable to pretty much any of our mass spectrometers. It's a capability we've never really had before in terms of the speed and the accessibility of different types of samples.
Coupling that with the potential of what scanning SWOF can do means that we could have a different view of these samples and cover many more samples, enabling much better statistical analysis of the data and a much better understanding of what's really going on in systems.
Better understanding of cancer and cancer treatment, better understanding of drug development, better understanding of how to protect people from a well-being perspective.
We're definitely in a world where we always need to quantitate, and we're definitely part of the organization that probably offers the best solution with respect to that. Not just today, but systematically over the many years where we've been introducing mass spec. It's at our core, and it's definitely an area where we excel at.
SCIEX will always be a leader. We have some of the best scientists in the world and engineers working to develop the next greatest thing, and I think we'll always be that way.